Okay, so welcome back to this session on carbohydrates, and we are here discussing on polysaccharides. Polysaccharides forms the most complex among the carbohydrates, and most carbohydrates present in nature they occur as polysaccharides. Oligosaccharides they are actually not so abundant among the oligosaccharides. Disaccharides are further frequently found. Okay, polysaccharides are also generally referred as glycans, and the most predominant monosaccharide found in polysaccharides it is D-glucose. So it is the D-glucose form which actually forms the monomer for the formation of polysaccharides. Depending upon the what you call the composition, there are two kinds of polysaccharides, that is homopolysaccharide and heteropolysaccharide. Homopolysaccharide, where you can see these are the two homopolysaccharides, and this yellow colored like glucose units. Okay, fine. So these are actually the similar molecules. You can see when it is hydrolyzed, it forms similar kind of molecules, right? It gives similar. Again, over here, this is just the differences. It is branched, but the monomeric units are all the same. Fine. So homopolysaccharides, it is when the polysaccharides are composed of a single monosaccharide building blocks. Okay, it is composed of the same type of monosaccharide. There are two forms or two kind of homopolysaccharides which we can identify or recognize. That is, one is which takes up the storage function, and the second is a structural as a structural element. The storage forms of homopolysaccharides include the glycogen and starch. So the structure we'll be learning in detail in the coming slides. Glycogen and starch both being a polymer of glucose. Okay, then structural elements, cellulose and chitin. Similar units get recurred to form the polysaccharide. That is what here referred to. Okay, it needn't be monosaccharides alone, but it can be modified monosaccharide as well. Okay, now in the case of heteropolysaccharides, polysaccharides when it is hydrolyzed, it forms like more than one type of monosaccharides may come into being. Okay, peptidoglycan, it is composed of more than one type of monosaccharide. Okay, so here you can see this is one type of polysaccharide where you can see two different types of monomeric units are there. That is, two different types of monosaccharide units bound together by glycosidic bonds form the heteropolysaccharide here. Okay, blue and the orange, it actually represents two different kinds of monosaccharides. What about here? We have three kinds, isn't it? 1, 2, and 3 here. Okay, so this is getting repeated, but each of the unit we can see it is formed of three different kinds of monosaccharides, multiple monomeric units. Okay, and furthermore, it is also branched. Now, so we'll be learning about the glycogen in detail. Glycogen, it is a main storage polysaccharide of animal cells, and it is especially abundant in the liver. And it is also found in muscle cells as well. It is a homopolymer of glucose. So it is a homopolysaccharide. It is composed of glucose units, especially the D-glucose. As already mentioned, when we are speaking about the monosaccharides, we saw that there are two kinds, D forms and L forms, and D forms actually it is being used in the formation of polysaccharides or oligosaccharides. Okay, they are more abundantly used up in the biological world. Okay, so it is D-glucose. It is the homopolymer. Okay, so here what happens is, okay, so glycogen, we can say this is how it is formed. Okay, it is a branched structure. So a polymer of alpha 1,4 linked subunits of glucose with alpha 1,6 linked branches. Okay, so these two are formed, these two kinds of what you call chains are formed. It is not a single chain, but this is how it will be. Okay, we can say this is being taken from another web page. Okay, so here you can see this is one chain. Okay, here there is a branching, right? And here there is a branching, yet another branching, and likewise glycogen structure, it is actually branched structure. It is having a branched structure, and the straight chains there, the monosaccharide units are connected by alpha 1,4 glycosidic bond. Okay, in the straight chains it is connected by alpha 1,4 glycosidic bond, just like we have seen in the case of maltose. Okay, two D-glucose units connected by alpha 1,4 glycosidic bond. But in the branched regions, branching points, okay, here at these branching points, these two glucose units, you can see if you are starting from, okay, this is the reducing end. Okay, so these are the non-reducing ends. So what happens is the alpha 1 carbon, what you call the C1 of this particular unit of this glucose is bound with the fourth carbon of this sugar. Okay, the first carbon of this sugar is bound with the fourth carbon of this one. So what happens here is, like, it is like almost like continuous. Fine. At the branching points, there is a change over here. Here, what happens is there is the alpha one, what you call carbon of this one is bound to the sixth carbon of this one. Okay. So here, what happens? This particular glucose molecule, that is this one at the branching point. Okay. This particular glucose molecule has the fourth carbon forming a glycosidic bond with the previous one. The first carbon forming a 1,4 glycosidic bond with the next one. At the same time, the sixth carbon is also forming a glycosidic bond with another glucose molecule. I hope it is clear. Okay. So at this point, it is alpha 1 6. It is in this direction. Okay. Here it is alpha 1 4, alpha 1 4. It is alpha 1 6. Okay. So it is alpha 1 6 glycosidic bond. I hope it is clear. Okay. So at all these positions, it is like that. So two kinds of bonds are found in glycogen structure. Alpha 1 4 along the straight chain, and wherever there is a branching point, it is alpha 1 6. Okay. So here I hope it is clear with this. Okay. So this is a straight chain of glucose molecule where one is a reducing end in the sense where so this is the reducing end over here. You can see why it is sorry, what you call non-reducing end. Why is it non-reducing? Because here the anomeric carbon is not free. Okay, and this is a reducing end where the straight polysaccharide chain or the glucose chain actually it ends in a free anomeric carbon. So this is a reducing end. Clear? Okay, so for every straight chain you can see there is a non-reducing end and a reducing end, and it is a reducing end which is ready for oxidation. Okay, it is here the oxidation happens, while this is actually resistant to any oxidation process. Okay, now at all the branching points you can see the sixth carbon. Okay, so if there is a branching over here, you can see this is the sixth carbon, the first carbon, second, third, fourth, fifth and sixth, isn't it? So this is the sixth carbon. The hydroxyl on the sixth carbon forms a covalent bond, a glycosidic bond with another glucose molecule. Okay, with the anomeric carbon of another glucose molecule over here. So this is how it happens. So wherever there is a branching point. Okay, so but there is a pattern. What happens there at every 8 to 12 residues there is a branching. That is how the like pattern is. Okay, so along a straight chain when we see, okay, so it is a straight chain, right? It is continuing from here, right? So every 8th or 8 to 12th what you call glucose unit, there will be a branching point. Okay, so that is how the frequency is, right? And glycogen, it is a very compact structure because at every 8th residue you can find a branching point. Okay, so it is very compact and it results from the from the coiling of the polymer chains, and this compactness allows large amounts of carbon energy to be stored in a small volume. Okay, so whenever there is a bonding between two monosaccharide units, water molecule is removed, isn't it? So it becomes more and more compact. So what happens is when we are comparing the number of glucose units stored in a glycogen structure against same number of glucose units in a free form, obviously in a glycogen it is more compact efficient. Okay, it saves space as well as it provides more amount of glucose molecules to be stored. So that is the efficiency of glycogen, the structure of glycogen. Okay, coming to starch, again starch is also another type of polysaccharide formed of glucose polymers. It contains two types of glucose polymer. We can say one is amylose and the second is amylopectin. Amylose is you can see it is straight chain, just like what we have seen here. Okay, so this is almost comparable to the amylose in the starch, and this is the amylopectin in the starch. Okay, so that is how the structure will be. Amylose, it consists of long unbranched chains of D-glucose residues. They are connected by alpha 1,4 linkages, just like as we have seen in the case of glycogen. And such chains vary in molecular weight from a few thousand to more than a million. So straight chains, it can be very much long. So we can't say that how many molecules can combine to form this amylose chain. Okay. Now amylopectin, it can also have a high molecular weight up to 200 million, but unlike amylose, it is highly branched. Okay, the glycosidic linkages joining successive glucose residues in amylopectin chains are alpha 1,4, and at the branch points it is alpha 1,6. Okay, and branch points it occur at every 24 to 30 residues. So when we compare glycogen with starch, glycogen is a little more compact. Why is it so? Because you can see at every eighth residue there is a branching point. But what about starch? Only at 24th residue you can find a branching point. So it is not so compact. Okay, so as compared with glycogen, starch is a little more less denser molecule or compound. Okay, so this is a structure. Okay, so this is the amylopectin branching at every 24th residue. Okay, and it is through alpha 1,6. This is alpha 1,6. Okay, so this is a anomeric carbon of this particular glucose molecule, right? And it forms a glycosidic bond with the sixth carbon of this one. Okay, so this is amylose and this is the amylopectin branched form. Okay, so these two together form the starch. Okay, so I hope it is clear. This is about the polysaccharides. We saw what is polysaccharide made up of, and the major two types of polysaccharides, that is glycogen and starch, and the difference between them, the structure, etc. Okay, fine. Okay, thank you.